Arrival of the action potential arrives at the synaptic impulse. Calcium channels in the membrane open, allowing calcium to move in the cell. The increase in calcium causes synaptic vesicles filled with acetylcholine, also known as ACH, which is signified by the orange nerds, to fuse with the membrane. ACH is then released from the synaptic knob by exocytosis into the synaptic cleft. The ACH is then picked up by receptors located on the motor end plate of skeletal muscle. Sodium, or Na+, represented by the purple Play-Doh, and potassium, or K+, represented by the blue nerds, channels open, allowing sodium ions to enter the muscle fiber. This increase in positive ions inside the muscle fiber initiates an action potential on the sarcolemma. Acetylcholinesterase in the synaptic cleft destroys ACH so that another muscle action potential does not arise unless more ACH is released from the motor neuron. This action potential travels down the sarcolemma into invaginations called T-tubules and stimulates the release of calcium, shown by the green nerds, from the terminal cistern of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin, signified by the red Play-Doh, into the sarcoplasm of the muscle cell. Calcium floods over the thin filaments and binds to troponin. The calcium that is binded with the troponin, signified by the green nerds and the pink Play-Doh, has pulled the tropomyosin off of the active site on the actin. High-energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the low energy state, releasing ADP, shown here by the blue Play-Doh, and moving the thin filaments over the thick filaments. This is known as the power stroke. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium then deattaches from the troponin, allowing the tropomyosin to bind back to the thin filament. Calcium is then pumped into the sarcoplasmic reticulum and attached to calsequestrin. The tropomyosin, now binded back to the active site, results in the relaxation of the muscle.